Welcome back. This is lesson 4 of the BVCN basic Unix tutorial coming from the Happy Belly Bioinformatics Unix tutorial, hands-on demonstration. So this is unit 4. We're going to be talking about 6 glorious commands that really somewhat show off the power of Unix and the types of commands that are built into our Unix system. Right, so these are going to be tools that you'll see, and you'll use them in varying levels of frequency, but they will give you an opportunity to see the power that is available here. And while we'll be going to be working with smaller file sizes, maybe only a couple lines with a couple of columns, you can imagine how this might be useful going forward with files that might be thousands of lines long with hundreds of columns. That you can then use to quickly parse and get to information that you're interested in going forward as part of this. One of the things I think is really interesting, particularly about this setup, is that the commands that Mike has listed here as part of his 6 glorious commands, I would say half of them are commands that I wouldn't even think about on a daily basis myself. And it's more to do with the way that Mike and I have come into bioinformatics from at different points in times in our career. And I think it really adds a testament to the fact that in bioinformatics, with tools like this, that there are very rarely the right way to do it, but there are lots of good ways of doing it, right? So there are ways of approaching almost all of these questions, not even with just the tools that are being presented today, but with other tools that exist out there. And the great thing is that nobody's wrong. Everybody is right in their own way, which is very utopian. And I think one of the many reasons why people really like open access when they're in computer science is that there just isn't a wrong way. There are ways that are slightly better, but even then those elements are kind of moot in examples like this. So let's start forward here. Let's move forward. Let's all make sure that we are in the correct starting spot, which is our usual starting spot. This CD squiggly Unix underscore intro. If you had just started up the binder because you are coming back to this like I was, that means that you're going to be in the right spot. Otherwise, we want to make sure we're in the right directory if we're continuing on from yesterday. And so what we're really going to be dealing with in this particular example, this lesson, is essentially the contents of an Excel spreadsheet. You can see here that we're going to be working with a file that's called gene_annotations.tsv. We know TSV stands for tab separated values. And so we'll be able to take this is what a quick look of what this looks like. So we have one column with a gene ID, which is just a sequential number counting up. The genome it's coming from, which looks like to be a strain ID, the co-ID, so this is related to KEGG ontology. If that's something that's interesting you, check back into functional annotation and a couple lessons. Lessons 4 or 5 might cover KEGG ontologies. And then from KEGG ontologies, you actually can get a KEGG ontology annotation. So this would actually be a functional component of the biology that you might be interested in being part of. And so to get to that point, to work with that file, we're going to move into the correct directory that we want to work with. And so we know that if we start here, we have 3 directories that we can go into: data, experiment, 6 commands. We're going to CD into the 6 commands using the tab complete to get to that next spot, and then hit enter there. And so Mike jumps right into well, let's take a look at these files the way that we want to, but we can also ls and see that there's a large number of files here that we're looking into, including the one that we're trying to target, which is geneannotations.tsv. And so we're going to follow along here. We're going to look at head, which is going to print out our first 10 lines of annotations.tsv. And that's going to give us our full contents. Well, not full. They'll give us the first 10 lines of the contents of the file. And then if we wanted to, like yesterday, we learned that we can modify head. Yesterday, in other videos that you may have watched just minutes ago, we learned that you can modify head by adjusting the number of lines that we're looking for. And so if we do head -n1 gene_annotations.tsv, this will just return back our header line, right? The first line of information that happens to contain all of our, what the various columns are representing. And we can use the tool word count that we learned in the lessons previously. We can do word count, gene annotations to see how long actually this file is. And there's 101 gene annotations in this file. So if we're including the header line on that one, that gives us the column information. It seems that there's probably 100 lines of gene information. And so the first command that we're going to work with is the command cut. And so cut is this, allows us to essentially take the columns that we are working with here and designate which columns we want to then move forward with going forward. So in this example, what we're going to do here is we're going to do cut. We're going to give it the flag F1. And so F here is short for field. And so we're counting up from 1. So this would be 1, 2, 3, 4. And if we then do that with the TSV at the end, you'll see that what we get coming out of that is the first column, which we know is the gene ID. So it's keeping the header line, and then prints out all of those numbers that are counting up sequentially for us up to 100. And so we can do that and not get as large of an output if we put in the, if we pipe it to head. So we can see our first 10 outputs, which include our character, our header line up here. And so we can modify this in a couple different ways to get different outputs out. And we'll keep using head here to make sure that we don't print out everything like we previously did and run off the edge of the screen. There are a couple ways that we want to interact with the file here. So the first example here is if we put if we designate 2 different columns. So here we're going to designate column 1 and 3 and designate it with a comma in between the 2. So we can run that. Again, we're going to leave the head there just to look at that. And so this is going to print out columns 1 and 3. And I can prove that to you. Let's just do that here. Head dash N1 gene annotations TSV, right? So column 1 is gene ID, column 3 is co-ID, right? And so we've asked it here, print 1 and 3. If we go back up, if you're following along, back to this line where it says cut dash F, and we change the comma to a dash, we're going to get a different output. And this different output now is giving us the information between columns 1 and 3. So column 1 was gene ID, column 2 was genome, column 3 was co-ID. By putting this dash, we're now capturing columns 1, 2, and 3. And so that's one of the main ways that we interact with cut is by designating which columns and how we want to integrate those columns together. And you can mix and match how this works, right? So we can ask for 1, 4, which will give us column 1 and column 4. We can say 1 through 3, and if there were more columns, we could do 1 through 3, comma, and then some other number of columns on top of that, right? So there's a lot of power here of selecting the columns that you want out of, subsetting it out of maybe a much larger spreadsheet that you're working with. And so one of the things that's really interesting here is this example that we can use. So there's a second version of this file in the directory called example gene annotation CSV. And so it's the exact same file that we're working with, but instead of a tab separated value, we're using a comma separated value. And so we're going to run the exact same command that we just ran here. It looks exactly identical to the file that we're working with, except that instead of tabs like this, there are commas. So let's try and run that command with the commas, or the file that has commas in it, sorry. So that's our example. We can tab that out because it's the only file in there with that name. And if we hit head, we're going to get everything, right? We get all 4 columns, 1, 2, 3, 4. And that's interesting in a way, because you're like, okay, well, I specifically asked for 1 and 3. Why is that the case? Well, one of the things that's interesting about cut is that by default, the delimiter that it's looking for is tab. So in this instance, our file consists of a line that is only separated by the commas. It's just saying, cut is looking at this and saying, there's only 1 column here. If I'm looking for the tab, there is only 1 column because that's what I'm looking for. The commas don't mean anything to me. So if we want to switch cut to look for commas and use that as a way of tracking that information for us, we need to tell it with a different flag, a second flag, to look for the comma as the delimiter. So we can do that with cut -D. Here we're going to do open double quotes, comma, close double quotes, -F, and then 1-3 example head. And so now this version that we're going to capture should look much more similar to this output as opposed to this output. And there we get it. We get 1, 2, 3 columns, and we're capturing them with the comma separated values. So one of the examples that we're going to do right here is this quick practice that Mike has laid out. And so we want to target just 2 columns, the one that's called gene ID and the one that's called co-annotation. And then we want to put it into a new file that's called IDs_and_annotations.tsv. Okay, so we should think about how we're going to approach this. We're going to use our TSV file, which by default has tabs in it, which means we can use the default setting of cut. We need to figure out what columns those 2 names correspond with. And then we need to use our redirect from lesson 3, where we're going to take that output and put it into a new file name. Okay, so let's figure the first step out is what columns are gene ID and co-annotation. And so that looks like to be, if we do this again, we can do, there we go, head. Getting a little spike in my connectivity. There we go. Right, so these are our possible column names. And so we want gene ID, which is 1, and co-annotation, which is 4. And so we can use cut -F1, comma 4 for our gene annotations.tsv. And now we want to capture that information. We can redirect that using the one caret director into a file called IDs_and_annotations.tsv. And now if we take a look at it, we look at the solution, that's what we want. Look, he did exactly the same way. We wanted to make sure which ones we were looking at. So nice. And so we can take a quick peek as to what that might look like by looking in our IDs file. And that's going to print out our 2 columns that we're looking at, gene ID and co-annotation. And then if we want to make sure that everything's working properly, we can double check that we've got all the information, which we can do with a word count. And it says 101, which matches what we did up above. Great. So the second command that we're going to learn in this series of 6 is grep, which is an abbreviation for global regular expression. And so regular expressions are ways of looking at strings of characters and recognizing the patterns that are part of them. And then in a lot of instances, once we recognize a pattern, we want to do something with them. And so there's a couple of arguments that we're going to use here. The first in the positional arguments is the string that we're going to be looking for. And then the second argument is actually going to be the file that we're going to draw from. There are other ways. Grep has some other levels to this, right? So we can use, once we see how grep works, you can actually use it to interact with files and files, text and files. It interacts in a lot of different ways. But let's do this version first. So we're going to look in a file. I'm going to hit clear because we're starting a new set of commands. So I'm going to look in the file colors. And it's colors.txt. There's 2 versions of colors here, but we'll get to that in a second. And if we hit head, we'll see it just lists a number of colors here. The number is less than 10, right? So we know that we're seeing the whole file. It's red, orange, yellow, green, blue, indigo, violet. And so if we wanted to look inside or use grep to find something inside this file, we can use it to find, does this file contain the word blue, something that we know it does. We can set it up like that, and we hit enter, and it will return. And so here in the binder, it's going to return a very easy way of denoting that we actually found what we're looking for. The color is going to highlight here. Maybe on your local system that might not be the case. Those are kind of somewhat settings that may or may not be preset as part of your computer or not. But here you can see that we've now, we went into this file, we looked, did anything match these 4 letters, the word blue, and it said yes. And so we can do this with multiple lines. So here we're going to look for just the 2-letter command RE. You can ignore right now the quotes. That was me in a habit that I have of mine of putting words that I'm looking for in grep in quotes. You see that it doesn't impact this. I'm going to do re here without quotes, and it will have no impact on how the program works. And so here it's grabbed lines that the 2 letters RE are next to each other, which include the word red and the word green. And so we can continue to work along with this. The next example is, how about if we try and grep the word black from the document colors? And so we assume that this isn't going to work because the word black is not part of the file that we're working with. And if we hit enter, we'll see that nothing gets printed. Okay, that makes sense, right? We weren't expecting black to be there, so it didn't find the word black. So let's go back to our gene annotations file, which has information about our KEGG ontologies in our 3rd and 4th columns. So again, we will repeat this step where we look at the first couple rows. Right, so these are our 4 rows that we're looking at. And so there are a number of a very large number, 22,000 KEGG ontologies to be exact, but all representing somewhat of a different functional component of metabolism. And so if we were to go to the Co website, we could look for this enzyme, epoxy quinosine reductase, that then has 2 co-IDs that are related to it. And so we can check to see in that annotation file that we have, do any of the annotations have this KEGG ontology identifier as part of them? And so we can do grep. And again, so this is, before we were using words or letters that were corresponding with each other. But here we can use a series of, let me make sure it's a 0. This is 0, great. We're using a series of, a string of numbers here, right? So it's K09765. We're going to pull that, try to see if we can see that in the annotations file. That version of epoxy quinosine reductase doesn't seem to be present, but I would assume Mike wouldn't lead us down the wrong path, that if we do this for the next file, yeah, great. So there are actually 2 matches to this KEGG ontology in the output that we're working with, this gene annotations file. And so there are, as it seems to be, 2 of them. And so we can do this step, right, where we're only targeting these 2 lines that happen to have the word or string of characters that match what we're searching for, right? So there were 101 lines, right? We've only pulled out the 2 that we're interested in. And we can combine that with grep and cut to print out just the 2nd column that have these K18979 matches, right? And so you can see here, column 2 is this column here, and it lists the genome IDs that we're working with, and will give us the 2, return the 2 genome names that we get from this match right here. So if we use grep, we can use the exact same command that we used before. I'll just hit up to bring it up again, and then we want to pipe this to cut, and we want to get just the 2nd column, so F2. And there you go. So now we've just printed the 2nd column using our combined first 2 tools. And so, as I mentioned at the beginning and is reiterated here, this is just like the very surface of what grep can do. It's worth noting here that we can add a flag, the -C flag for count, before the word that we're using to search for, the argument that we're using to search for. So if we did grep -C, and it did K18979, gene_annotations.tsv. Instead of getting the full line output, we get a number. And it's just counting for us how many of those are occurring in our file that we're working with. Okay, so let's move on. This is the 3rd command of our 6, which is the command paste. And so paste and cut are both working on columns. And so if cut was removing columns based off of the delimiter tab, paste is adding columns based on the delimiter tab as well. So we're going to look into this file that's here. That's part of this already. which is head colores, which is colors in Spanish, right? And so we have the Spanish counterparts of the English version of the colors. So you can look here, if you are familiar with Spanish, this is actually in the same order. And so what we can do here is we can take paste, and we can paste these two files together. And so based off of the description, what it should happen is we should take our file colors and our file colores, and we should put these two files together with a tab between the two files. So we can do paste, colors.text, and then colores.text. And there you go. So what we've done here is taken our two files, and now we've stitched them together with a tab in between. We're going to talk about this in a second, but keep in mind that these two files had the order that we wanted them to be in. All right, so let's look at this file, this new file that we're going to start working with, and take a look at it. It's called genesandseeks.tsv to get an idea of what might be in there. Genesandseeks.tsv. Oh boy, look at that. So this appears to be a tab delimited file that is storing protein annotation information. So here it has a gene ID, an amino acid length, and then the sequence. And then because the sequence is extremely long, right, it runs onto the next line and continues down until it stops. And then the next line starts that has a 2 here. And so this is a little messy, and we want to make sure that this isn't some artifact of any returns or options that might be hiding the fact that this is not one contiguous line. And so one of the things that we can do is we can use less with the -s option, which will remove this line wrapping that's happening. And so that'll give us at least some confidence that what we're looking at is just one contiguous line, and we don't have some superfluous amount of returns there. So let's do less-S, and the capital S is important there because that's the flag it's looking for. Lowercase s is a different flag, and we hit enter. Right, great. So we can see that it is in fact, that sequence does run off the edge of the screen there. And so the information that we want is in one line, and we can treat this just like any other tab delimited file. Remember to get out of less, we're going to hit the letter Q. That's going to escape out. You can still see that this information is here because we used head on that one. And so these gene IDs are counting up in the same way that our other file that we were working with gene annotations was counting up. And so we're going to hope, and Mike has done a very good job of this, that it's counting 1 through 100, and these 1 through 100 are all related to each other in the same way. And we're going to use paste to stick our annotations, which have gene IDs, and our genes and sequences together into one file. So we're going to do paste genes. Oh, sorry, gene underscore annotations is the first one. .tsv, and then genes seeks tsv. And we can check to make sure that that's happening correctly before printing it out to the screen by using head -n1. And so now what we're getting, we get our first 4 columns that we've been getting for a while: gene ID, genome, co-ID, co-annotation. And now we have the 3 new columns that we're working with: gene ID, amino acid length, and sequence. And then Mike has a warning just in case you got an error here and it's still printed. This has to do with the way that paste and head are interacting with each other. And in this situation, because head is only asking for 1, it finishes faster than paste can do its job. Okay, so if we wanted to, we could take everything except for the 5th column, which is a repeat column, right? So gene ID here and gene ID here are the same. And we can combine all of the things that we've learned to this point, not all of them, we're not using grep, to only print out the novel information and not to retain the second gene ID. And so here is where we're getting into that, what we were talking before, where we can use a mix of notations in cut with the fields that we're looking for. So we're looking for fields 1 through 4, and then we're going to put a comma here. We're skipping 5. So by not including it, it won't be included in our output. And then here we're using 6 with a dash, and that's implying that we're going to go from 6 until the very end, right? So we could be explicit and say 6 and then have the number of the columns that go there, right? So that would be column 6 and column 7. But this is another way of doing the notation. And so we can actually, I can show a couple ways where that will work the same way. So let's do paste gene annotations.tsv, genes, TSV. We're going to do a pipe to a cut. We're going to do -F. We're going to follow along. We're going to do columns 1 through 4, skip 5, column 6 with a dash, and then head -N2. Okay, so now when we hit enter, so we're asking for the first 2 lines. We get exactly what we wanted here. You see, I got one of those little errors here that suggests that the whole process hadn't finished completing at that point. But what we get is our columns that we wanted, our first 4, 1, 2, 3, 4. And then instead of the gene ID that's been dropped, we now have the amino acid length, which is right here, and then the sequence that goes on from there. And so I mentioned we can actually do this in a couple of different ways. So let's just explore that, right? So we can do 6-7, right? So that's telling it, that's being explicit. We're saying capture amino acid length and sequence. And so this should print out exactly the same content, which is good. And because these are sequential, 6 and 7, we could also put a comma here if we wanted to, right? We could say 6 and 7, not 6 to 7. And this would also generate the same output, also with the broken pipe information. And so what you can see is that cut interaction, the way that we're writing cut to on the screen, we can actually come up with multiple ways to do exactly the same thing that we were planning on doing in the first place. And so this is one of those things I wanted to note here. In both examples, right, we were working with under the assumption that the files that we were working with were exactly the same order. If they hadn't been, and we had used paste, we would have just artificially stuck 2 files together, and paste would have not cared 1 iota if any of the columns actually were related to each other. And so some of the commands that we aren't going to cover with, and you might see later as you're working with Unix, include the command sort, which does exactly what you would think it does, and the command com. Okay, so let's move on. This is now our 4th amazing, fantastic new command that we're going to learn. I'm going to hit clear here to get out of this. So this one's called sed, which is an abbreviation of stream editor. And so this is where if grep was looking for pattern matches, sed looks for pattern matches, and then also allows you to make changes based off of that pattern, which is a very, very useful way of working with this. And so one of these things, Mike is using a real world example here that you can read about the drama of having to rename instances of their genes for their new genome that they were submitting to NCBI. But that's an anecdote that you can read on your own. But what we can do is we can use tail here to look at the bottom part of gene _annotations.tsv. And so Mike is trying to emphasize that we have here at the bottom, we have information related to our new genome, which has this annotation, or this name UW179A, and that we actually want to change this now from UW179A to UW277. So we're going to use sed, and we're going to follow what's written here. So we're going to do sed space. Here's some important ways of structuring this correctly. We're going to use a single quote, open single quote here. We're going to put an S, a /, the contents that we are looking for, which is UW179A, another /, and then what we want to change it to, which is UW277. And then we need to close it with our /, close it with our single quote, and then gene annotations.tsv tail. Okay, let's take a look and see what happens here. Oh, nothing happened here. Now that's a particularly interesting thing. Oh no, sorry, a lot of things happened here. Wow, let's make sure we cut that. Here, what we did was that we actually, you can see we can change UW179A to UW277 everywhere that it was before, right? So this was in the file. We were able to make those changes, and so I was reading it out to it as I went. But there are 4 major components to this, which we use 3 of in this example. right? The first is this S before the first slash, which is short for substitute. The second is the element that we're looking for, and then the element that we're trying to replace it with. And so currently that fourth spot, which would happen here between this forward slash and this single quote, isn't currently being used right now. So now that we've done this preview with tail, we can actually store this new information in a new file. So we're going to just hit up to bring up our last command, and I'm going to replace pipe tail with a redirect into a file that we're going to call modified. Spell that right. Gene annotations.tsv. And now if we hit tail in that modified file, which I can tab complete, you can see it looks exactly like that tail that we were looking at before, which is great. And is different than the original tail of gene annotations.tsv. Okay, so one thing to know about sed is that by default, it only changes the first occurrence of something in the line, right? So this is why it worked for what we were working with here. There's only one occurrence of UWA 17.9a in each line. So the first time it sees it, it makes the change. So if we were to do this again, so we're going to do head gene annotations with a different example. So there's a number of lines here that have the of NA not available for both the co-ID and co-annotation. And so what we want to do is we want to use sed to change all of our NAs to NAs with some carets around them like this. And so let's try this command and we'll see what happens when we do that. I can assure you it's not what we're going to expect. So we're going to do sed single quote s /NA, which is what we're looking for. And then we're going to do a less than symbol, NA greater than symbol / single quote, looking in gene annotations.tsv. And now we're going to use head to look at the same first 10 lines. And so what you'll see here is because sed only works on the first instance of everything it finds, it has effectively changed NA in that first column, but it has not changed NA in the second column. And the way we fix that is by using that fourth position that I mentioned just a second ago, where if we hit up, and in between this / and this single quote, we put a G, and now we hit enter. The G stands for global. And so now what will happen is that everything that has NA in all instances in the line get replaced. And you can see that it might not actually function the way that we exactly want it to, because everywhere where there was a DNA, including lines that didn't have NA in it, have now been also replaced with this caret structure, right? So it's one of those things that we need to be somewhat careful with. When we're doing these types of substitutions, because something like this might happen where we're actually trying to target this NA here, but all of the NAs might be substituted somewhere else, which can always be problematic. The next tool that we're going to learn about is awk. And so awk is a way of filtering based off of contents of a column, and then also has some built-in ability to do calculations. And so we're going to use awk on a different file. We're going to use a blast output here. This is our 5th command. I'm going to hit clear to clear everything out here. So we're going to look at this file that's sitting in our 6 commands directory called blast_output. So this is an example of a blast output. So blast being a liner that you can use to search for local alignments between sequences and then get some scoring information about how that works, just in case you're unfamiliar with that. If you are unfamiliar with that, the next lesson, lesson 2 of functional annotation, will cover blast, which will be out shortly. And so we can use awk, like I said, to filter things based off of cutoffs that we want to use for our columns. And so in this blast output, we have our query ID, our query length, our subject ID, our subject length, and then % identity, and then alignment length. And so awk uses a similar annotation to cut, where we're counting how many up we are. So this is column 1, 2, 3, 4, 5. And so we might be interested in, from this file, capturing every instance where our % identity is really high, so above 95%. right? And so we can do that using this annotation that we're using awk here, whereas there's single quote space, $5, which is telling us treat 5 as some type of variable that you want to look at, but look at all elements in that column 5, and then return anything that's greater than 95. Another space, and then we're using our input as the blast output.tsv. So if I do awk, single quote open with a space, $5, space here, just to make it easier to read with a greater than, 95, space, single quote, blast output. So now this should return all instances greater than 95, which in this example it does. And you can see we've excluded these ones that were less than 95, so in these in the high 80s, and we only captured 100, 96, 96, 195. And so you can, one of the things that is noted here is that awk is looking for a number in this information in column 5. And so when it finds a row that has only text information, it doesn't actually know how to make that comparison and assumes that that's just something that you also want to be output as well. So we actually capture the header line here with the information that we want. And then we get only the rows that have in that column greater than 95. And so we can use multiple ways of tackling this, right? We can use awk to add layers and layers on top of what we want to do. And so in this instance, what we're going to do was we're going to use the and symbols of awk, which is represented by 2 ampersands together. This is in contrast to the or sign, which is represented by 2 pipes together. And we're going to say, hey, we want all rows that are greater than 95% identity, but we also want where the query length, this column, is greater than 1,000. And so if we're looking at this output here, there should only be 2 rows that are greater than 1,000, these ones right here. And so we can do this and command as part of awk, where we do awk, single quote space, $5 greater than 95, 2 ampersands, and then a $2 greater than 1,000, space, single quote, and then blast output.text. And so again, because we gave it a number, awk is not looking to filter these text rows, but then it finds the 2 rows that we were hoping for that are greater than 1,000. And so it was mentioned before that awk is also able to do small amounts of calculations. And so one of the things that can be very common in a blast output is that you can get a really high identity over a very short length of a very long sequence, right? And so this is generally, makes some sense in the biochemistry world, but might not be what we're looking for trying to identify proteins that look very similar to each other across their entire length. And so what we can do here is we can perform a little bit of math to go along with that. And so what we're asking here awk to do is what we've been doing at the beginning, find all columns where 5 is greater than 95. And then we want to find every instance where our 6th column, which is our alignment column, is greater than our query column times 0.9, so 90% of the length. So we want to capture only instances where the alignment length is not at least 90% of the query length. And so we can type that all in. Awk, single quote open, space, $5, greater than 95, 2 ampersands. We're going to do $6 is greater than $2 times 0.9. So that's the percentage that we're using here. Blast. All right. So this is going to give our output. So it's every time that it's greater than 95, and then every time that our alignment length, this number, is greater than 90% of the query length. In this instance, there are 100%, but you can see now that it's, instead of looking at just things over 1,000, which returned 2 lines, we removed this line here, which was short in terms of its alignment length, and we captured instead not things based off of their absolute alignment, but their relative alignment to the query, which is pretty exciting. And then lastly, the last command that we're going to work through is tr, which is short for translate. And this is fortunately very, very short in terms of what we're looking for here. And so One of the things that becomes pretty, can be somewhat frustrating, and it's something that got mentioned yesterday in lesson 3, is that when you work with some types of Excel or Microsoft Office programs, they replace what is the very common new line indicator of \n with a specific Microsoft version, which is a \r. And so when we export an Excel file that doesn't correct for this, and so something that I've noticed recently is that newer versions of Excel may actually do this for you, which is actually pretty interesting. They seem to recognize that when you export something as a TSV, you don't want the Excel delimiter here, but or new line delimiter, but sometimes that's not the case. And when we look at a file that looks like this, so we're going to do gene annotations, and then there's another one that's called _Excel exported, right? If we do this and it has incorrect new line interpreters, what happens is we don't get new lines. Every place there's supposed to be a new line, it's interpreted by this ~m, which is an indicator that what we're looking for isn't accurate here. So this is a whole huge mess, right? We don't get the lines that we're expecting. We can use Q to get out of this less command here. And then if we do a word count of this file, because there are no new lines, there's nothing registered here, right? Because there's no actual return character for a new line, which is what word count new line is counting, it doesn't find any and returns a 0, even though we know there's content to this file. And so we can use this translate command, this tr command, to replace these Excel return commands with the Unix new line command. And this is going to work a little differently than we're used to. Normally we would do tr. Maybe we would be some way of using this information here and here, and then reporting the file that we want to change. Here, what we're actually going to do is we're going to use a new redirector that actually reads in a file. So normally we read out a file, which uses the greater than sign. To read in a file, we use the less than sign. So we can do tr. We're going to do double quotes here to designate what we're working with. \r, close double quotes with a space, double quotes, \n, close quotes. And then we're going to read in our file, gene annotations _Excel. And then we're going to read out a new file that we're going to call gene annotations _fixed.tsv. And now when we look at the contents of gene annotations _fixed.tsv, we can see we get the output that we're expecting here without those ~m's that were prevalent when we were just looking at the Excel file. One of the things that's really interesting, and I think is pretty an interesting way of looking at this, is that we can also do this using sed from earlier in the commands. And so sed works the same way, right? Translate took one letter and found all instances of it and replaced it in our file. And if we did it with sed, we could do the substitution /. Here we're going to replace our Excel returns with our new lines globally from gene _annotations Excel. And here we can pipe it to less to take a look to get the output. And we get the same output as we got using translate. So it's just one of those elements, as we were discussing at the start of this session, is that there isn't a defined right way of doing this. There are lots of ways of tackling options to make sure that what you're presenting or what you're doing, it kind of fits what you are comfortable and what you learned, right? So both of those examples, both the translate and the sed, you could see maybe why you would want to use translate. It's a little shorter in its command, might be a little easier to remember than sed. But if you're using sed all the time and it comes like second nature after some period of months or years working with it, there's nothing wrong with doing that. And so to round out this lesson, let's go over the 6 commands that we learned. Cut, which cuts columns from tables. Grep, which finds matches and returns the lines that have those matches. Paste, which sticks columns together of files. So you just take files and you stick them together with tab columns. Sed, which is search and replace. Awk, which allows us to filter columns based on calculations and values that are present there. and then translate, which allows us to swap characters for other characters. So that concludes lesson 4. We will move on to lesson 5, which is variables and for loops in the Unix command line. Thanks for joining.